Hi, I'm Mrs. Jardine, and right now what we're going to do is we're going to talk about sponges. So first I'm going to review the characteristics of animals because this is the first animal that we're going to talk about. Do you remember any of the characteristics of animals? They're multicellular, which means every single animal in kingdom Animalia is made up of more than one cell. They're heterotrophic. Hetero means it eats other things. So every single animal in kingdom Animalia has to eat something in order to get energy. They have no cell walls, zero. Plants have cell walls, fungus have cell walls, and bacteria have cell walls. Animals do not. They have a few to a lot of specialized cells. And quick question, were you listening? Does every single animal have all of these characteristics? Yes, they do. All right, so I'm going to review protostomes versus deuterostomes. If you remember, protostomes, here, I'm going to move this. Here is the beginning of embryonic development. And gastrulation, what happens is the blastopore turns into the mouth, okay? And that's in protostomes. Deuterostomes, the blastopore turns into the anus. The blastopore, if you were wondering, or if you forgot, is a ball of cells. Kind of like if you take a tennis ball and you cut it in half, it's a hollow ball of cells. And so the blastopore is this opening right here, okay? So these are all different germ layers. So you have endoderm, mesoderm, and ectoderm. And so all these different germ layers, you can see here the mesoderm is blue. Down here, the mesoderm over here, it kind of looks like Mickey Mouse ears. Here the so you'll say this is the outer layer here, and this is the inner layer here. So this would be the ectoderm. Ectoderm turns into like the epidermis. This would be the endoderm that turns into the inside. So those are all the parts, and see how they both have that. And so in a protostome, the blastopore turns into the mouth, okay? Protostome, blastopore turns into the mouth, okay? In deuterostome, blastopore turns into the anus, okay? So deuterostome are like chordates, so like humans and mammals and all those vertebrates, backbones and whatnot, as well as echinoderms, so like starfish. Protostomes are like the invertebrates, like the bugs and things like that. So things, you would have to do that. Oops, I read the wrong side. Sorry. Okay, so what we're going to mainly talk about today is sponges. Sponges' habitat is the seafloor, obviously. And the first people to classify them thought they were plants, but they're not. This is a great video. I'm not going to show it on here. You can watch on your own. So this shows all the characteristics of animals, and it has a really good clip in there where they put dye around the outside of a sponge, and then you can see the yellow dye go into the sponge and come out the osculum. That's really cool. It's my favorite part. Okay, so the characteristics of sponges. Sponges are in phylum Porifera. Get it? They have pores, phylum Porifera. And sponges are the examples. There's lots of different types of sponges, but they just call them sponges. So they're sessile. What sessile means is that they don't move as an adult. Obviously, when the zygote is made, that flies through the water, swims, floats, whatever, through the water, and then that lands somewhere. And then when it grows, and then it doesn't move, but it does move when it's like the zygote stage. Zygote, if you forget, is a fertilized egg. But when once it attaches to the ocean floor, it doesn't move. So it's sessile. It doesn't have any tissues or organ systems. So like that ectoderm, endoderm that I was referring, it doesn't have any of that. It doesn't have organ systems either. It is asymmetrical. What that means, it is not even on both sides. So like this bear here, this is my kid's drawing. It just happened to be on the table. Is symmetrical. You can cut it down. Not drawing, it's cut picture. You can cut down the middle, and it's bilateral symmetry, if you remember that. The sheep, again, bilateral symmetry. Fish, earthworms, bilateral. Whereas a sponge is asymmetrical, which means you cut it down the middle, and it will not match up. Fold it in half, it's not going to match up. They have here, so here's a sponge right here. They have these little tiny cells. This is a flagella here, and this is choanocyte, which sits in there. So this top here is an osculum, and that is where the water goes out. So it goes in on the sides and up out the top, which I will show you later on too. Here, should I draw it? Want to see it? So it goes in these holes here. We'll draw blue because that's what colored water is always perceived at. So it goes in these holes, in these holes, in these holes. And so scientists in that video, what they did is they put a whole bunch of dye in the water outside the sponge here, okay? And they're like, oh, what's going to happen to this dye? Let's watch. And then they watched it all get sucked in. and then I can't whistle. My kids make fun of me because I can't whistle. And then it goes out. So it goes in through the pores in the sides of the sponge, and then it goes up out the top. So this hole in the top here, I should switch colors, but this hole right here is called an osculum. And see how it sits on the ocean floor. Okay, let me click. All right. So they're considered large water pumps because water is continuously circulated through there, and then you have these specialized cells that grab any food particles, like microscopic food particles, things floating in the water that the sponges would want to eat, and then they engulf them, and then they digest them, and they go through the sponge's body. That flagella on that cell I was showing you, it helps to move the water. So it's like, woo, 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 so the water goes up and out. That's how you spell osculum, if you're wondering. Osculum. So that's that top of the sponge that the water leaves through. Enters into the pores. So like right here. Out of the pore here, right here. So here's a pore. So water goes in through the pore. Okay, I don't know what's happening to my screen, but it's fine. In there, and then these are called collar cells. That's right there. See, it's called collar cell. There's the flagella, help to move the water out, so it doesn't just stay there. It just helps it to move it up. I don't know. Do you know what's happening? Not quite sure. So it's kind of cool. I'm gonna move this out of here. I'm gonna go back to an arrow. This is a amoeboid cell right there that would digest any food that would get absorbed. But so the water goes in, they absorb right away any food that would be in the water that they're able to absorb, and like take in. You know what an amoeba is? If you've ever taken say you take an egg, right? And then you have a frying pan, you crack the egg and it spreads all out. And say you fried potatoes in that pan first, and you took all the potatoes out because they were done, right? And oh no, there's one little tiny potato, and you're like, it's fine. You crack that egg, then the egg goes around the potato and it eats it. That's how amoebas eat. Because amoeba literally looks like a fried egg. This is amoeboid cell, see? Kind of looks like an egg cracked in a pan. It's every single one of these is an amoeboid cell, and they all look different. Every time you crack an egg in a pan, it looks different. And so if there's a piece of food particle right there, then they're going to engulf it. It's called phagocytosis. They're going to engulf it and eat it. Oh, yeah, food. Okay. So that's what they do. So water goes in through the pore. This is called a central cavity. You know why? Because it's in the center and it's cavity. And then the water goes up and out. So the central cavity whole purpose is to let the water go up. There you go. Now you know how sponges eat because they're animals. So they eat. They filter the water. They're filter feeders. Did I go over everything? Movement of water provides feeding. We went over that. Respiration. So they get, there's something called dissolved oxygen in the water. That's how fish get their oxygen from their gills. So sharks get their oxygen from their gills. And sponges need oxygen too because all animals need oxygen, right? And so they get it through the water. It's called dissolved oxygen. So they get that through their collar cells, circulation. They don't have a circulatory system. They don't have a heart. They don't have blood vessels because remember, they don't have any tissues. So they can't. They don't have lungs. They don't have gills because they don't have any tissues. Excretion. That just basically all goes in through their pores. They absorb what they need to, the food, the oxygen, and they excrete it out their osculum. They're very simple. That's why we learn about them first, because they're the most simple animal. There's something called spicules. Those are really cool. Look at these. This is a spicule under the microscope. Isn't that cool? So not all sponges have these, but some do. They have them as protection. So if something comes to eat it, they literally look like glass shards under the microscope. Then they're like, ow, that really hurt. Ah. Whatever animal that tried to eat it, and then they won't eat it again. The spicules are made from archaeocytes. I'm saying that correctly. The softer sponges are the ones that because you can buy sponges at a store, all natural sponge, right? People use them to like wash their cars or their bodies. Do you think those have spicules in them? They don't. No, because do you want to wash your body with shards of glass embedded in a sponge? No, you don't. But a lot of them do. So I like this picture here because it shows you. Okay, so here's the outside of this right here. This is a spicule. Can you see it? Right there. And they're all different shapes. If you Google up spicules, you'll see them. They're all different shapes. And then this is an amoeboid cell right here, which is behind this collar cell right here. And that's the flagella. And these are pores. So the pore goes in, or the water goes in the pore. I'm really bad at drawing. It does go back and forth. Sorry. And this is the central cavity. It's in the center. Out the osculum. Boom. It's not difficult. This is a cool picture because this shows you what I just tried to draw you. So the outside water goes in, it goes through the flagella, the collar cells, flagella have to move it, and then it goes up. The choanocytes are collar cells. I like to say collar cells because that's easier to say. They engulf the food particles because they have that amoeboid cell behind it. And archaeocytes, they help to complete the digestion. They get their oxygen from the dissolved oxygen in the water that diffuses in the cell. Did you guys remember what diffusion was? You remember? I'm going to run a different video. So if you have an upstairs in your house and you have a cake oven and say you bake a whole bunch cake, right? Cake smells really good. You open that door and you're like, oh, it smells so good. Or cookies or whatever your favorite thing is, that bread, cookies, cake. So in the kitchen, that smell is very concentrated. You're like, oh my gosh, it smells so good. And then maybe after like 10, 15 minutes, all of a sudden you're upstairs in your bedroom or your brother is, and they're like, what'd you make? You made chocolate chip cookies? And they come running downstairs. Well, you didn't tell them. How did they know? You didn't tell them you made chocolate chip cookies. You didn't say anything because you wanted to eat them all yourself. Because you're like, my brother's mean. I don't want to share. And what he found out through diffusion, because that smell, high concentration of smell in the kitchen, starts to diffuse out. It starts to spread out to low concentration everywhere else. Well, the smell is very, very low upstairs in your bedroom. So high concentration in the kitchen, low concentration in your bedroom. So then it diffuses from high to low. And once the whole house smells like chocolate chip cookies, it still diffuses. It still moves around. It's called dynamic equilibrium. It's still changing. Okay, so that's diffusion. So if there's a high concentration of diffusion in the water and low concentration in the cells, that there's a high concentration of oxygen in the water and low concentration in the cells, that oxygen is going to diffuse right into the cells. Carbon dioxide in any waste. Ammonia is a byproduct of digesting food. We change ours into nitrogenous waste, but like aquatic organisms usually have high concentration of ammonia because they can just excrete it right away because they're in the water. So that's why it says ammonia. It's like waste that would be like in your urine. We don't have ammonia. We dilute it. They don't have a nervous system because remember, they don't have any tissues. So they either use toxins or they use, they can be poisonous or they have spicules. The reproduction is sexual or asexual. They can make buds. So like part of, like if I was a sponge and this is the side of my sponge outsideness, I could turn this into like a bud. And then it can float off into the ocean and land somewhere else and turn into another sponge that looks like that has all the same DNA that if I were a sponge, would have. Or they can release sperm and egg into the water, and then they join up, and then they make a zygote, fertilized egg, and then they land, and then they make another sponge. Their sperm and egg go into, I said into the water, but I should have clarified. Sorry, it goes into another sponge. So the sperm would be released into the water, and then that would go into another sponge, and then it would fertilize the egg inside the sponge, and then it would be released. Goes to zygote to larva to sponge. Archaeocytes are the specified cells. So not only, they have lots of things going on. Not only do they help to finish digestion, they help to carry the sperm to the egg. So this is kind of a picture of budding here. Oh my goodness. So budding is what I told you, that part of the sponge breaks off. Gemmules are groups of archaeocytes surrounded by spicules. And what they do is, so say the sponge makes a gemmule. So this is a group of archaeocytes. Remember, archaeocytes can do they can do the digestion. Archaeocytes can help with bringing a sperm to an egg. They do a lot of things in the sponge. So they take some of those, okay? They surround them with spicules. Remember their protection. And they kind of hang out, and because if the waters are polluted or they're too cold or whatever, the temperatures aren't right, there's not enough, they can just hang out and kind of protect them. And then when the habitat becomes great again, the sponge is like, oh, okay, I'm going to come right back out. They don't think it because I'm a brain, but then they come out and then they kind of like hatch from their gemmule. They don't hatch because it's not an egg, but you know what I mean? And then they turn into a sponge. That's kind of cool. We can't do that. We just get sick. All right, so sponges have a symbiotic relationship with algae, bacteria, and plant-like protists. There's 3 different types of symbiotic relationships. I said this in the biology communities, I think, PowerPoint. Symbiosis is where 2 animals live closely together and at least one species benefits. So if the plant-like protists and the sponges are living together, then it would be a mutualistic relationship because they're both getting something out of it. If it's a plant-like protist, it's probably giving off oxygen, and a sponge gives off carbon dioxide. So then they're both getting that from each other. So that would be a mutualistic relationship. Coral does that too. All right, I'm done. So let me know if you have any questions, but that's sponges in a nutshell. I hope you learned something. Bye.